Okay, to begin, a couple of announcements. First, if you haven't voted yet, go get it done. You only have until tomorrow, so go get that done. It's really important. So use that franchise and go vote. The second thing I want to tell you about is we've taken a poll, we've taken a vote, and the poll closed at 10 o'clock today, and it was the overwhelming consensus that we should move the exam from Wednesday to Thursday. So we'll open the exam up on 1 o'clock on Thursday. We'll give you a little bit more time. And I was just a little bit concerned about people being distracted on Tuesday with the election. And so I think that it would be best if we can put that off another day. And the overwhelming number of students in the class agreed that Thursday's a better day all around. So we can do that easily enough with this online exam. So let's put it off until Thursday. What we're going to do today is I'm going to finish that story we began on Friday, which was the macrophages as antigen-presenting cells. And I'm going to go through the idea that the macrophages arrived at the site of bacterial proliferation, and the bacterial proliferation now has left a whole bunch of bacterial parts around. And those proteins of the bacteria can now be taken up by endocytosis, by the macrophages, passed through their lysosomal system, and then displayed out on the surface as an antigen fragment. Just a short antigen fragment, about 16 to 20 amino acids of one of those foreign proteins. This macrophage now will display that protein out on its surface on a protein called a major histocompatibility class II protein, or MHC2 protein. There are only 3 cell types in all of our body that are professional antigen-presenting cells, and these professional antigen-presenting cells are the macrophages, a cousin of them, the dendritic cells, which are probably actually a more important cell in the story. We'll use the macrophage as the main character. And then a series of cells we'll talk about later today called the naive B lymphocytes. So what we'll see here, one of the ideas I want you to hold on to is the macrophages, or in this would be the dendritic cells too, would provide a kind of bridge between the adaptive and innate immune responses. That the arrival of the monocytes at the site of bacterial proliferation, that is part of the non-specific or innate immune response. That arrival then allows those monocytes to mature into macrophages. The macrophages then can gobble up foreign proteins, and then they'll display those foreign proteins on their surface. They'll leave the site of bacterial proliferation and wander off into any number of germinal regions. And in the germinal regions, these antigen-presenting cells will present to other cells of the immune system. And that will help gear up the immune response. And we'll see a little bit of how this happens. But we'll see then that these macrophages and dendritic cells are a bridge between the innate immune response in the form of inflammation and the adaptive immune response in the form of proliferation of other immune cells. Then we're going to drill down into the cells of adaptive immunity, and we'll see that the adaptive immune cells come in 2 great categories. They come as B lymphocytes and T lymphocytes. The B lymphocytes are different categorically from the T lymphocytes. Now, we discussed the T lymphocytes on Friday, saying that they had to pass through the thymus in order to be selected. But the B lymphocytes are different creatures altogether. They leave the red bone marrow, then they go off into a germinal region where they mature. And these naive B lymphocytes, if they have a protein on their surface that recognizes a foreign protein called an antigen, then those cells will proliferate. Those proliferating cells then will eventually become something known as a plasma B lymphocyte. The plasma B lymphocytes secrete antibodies. They have this gigantic protein synthesis machinery factory that makes these antibodies. And those antibodies then are secreted, and they're secreted into the extracellular environment. Those antibodies will be specific for a particular antigen. They'll recognize a sequence of 16 to 20 amino acids on a foreign protein. Then those antibodies will bind to the antigen and form an antigen-antibody complex. And are going to produce antibodies, in the case of COVID, we think, are going to bind to the spike protein. and then bind to the spike protein and activate the spike protein so it can't enter cells. We think that's how these antibodies are going to work, and that's the main idea of generating vaccines. Most of the vaccines are aiming at that spike protein because that's the keys into the cell. And we'll see how forming an antigen-antibody complex can inactivate that particular protein, or more generally can inactivate any potential pathogen. So we'll walk our way through the humoral branch. We'll talk about the importance of the antibodies and their structure and how they get specificity. And then we'll talk about the cellular branch. The cellular branch has T lymphocytes. Again, these are the cells that, in order to be selected for potency and specificity, have to pass through the thymus. In fact, T stands for thymus. And the T lymphocytes come in two categories, two varieties. There's something known as a CD4 cell. The CD4 cell has another name. It's also known as a T helper cell. We'll see that the CD4 cells will recognize, using their T cell receptor, they will recognize antigen fragments presented on MHC2 proteins. So that tells you straight away, if we're dealing with MHC2 proteins, that tells us that the T helper cells are only going to be recognizing these professional antigen presenting cells. Could be dendritic cells, could be macrophages, could be naive B lymphocytes, but they're only going to be talking to those cells that express MHC2, and the MHC2 proteins, cells that express MHC2 proteins are in fact the professional antigen presenting cells. The other great category of T lymphocytes will be the CD8 cells. The CD8 cells have another name. They're called T killer cells. And these T killer cells, again, like the CD4 cells, like the T helper cells, they have a transmembrane protein called a T cell receptor. The T cell receptor has a particular three-dimensional shape that will fit precisely onto an antigen fragment. But it will only recognize antigen fragments presented on a particular transmembrane protein on other cells called MHC1, major histocompatibility class 1 proteins. So the T killer cells we're going to see are monitoring for foreign proteins expressed on the surface of all other cells except for erythrocytes. All cells that have nuclei will express these MHC1 proteins. And 99.999% of the time, what these MHC1 proteins are carrying is a variety of protein from inside the cell, a self protein. So those won't be recognized if the T killer cells are working properly. But if we have virus-infected cells that are generating proteins that are now being displayed on the surface of the cell, being displayed on the surface of the cell on the MHC1 proteins, then the T killer cells can recognize that foreign protein on the MHC1 protein and kill the virus-infected cell. And we'll see how that gets done. Before we get through all that, though, we need to spend a little bit of time on antibodies, where they come from, and how they gain their specificity. Again, the major idea here is that antibodies will be secreted into the humors. That's why it gets the name humoral branch. And these antibodies are secreted into the extracellular environment. The function of the antibodies is to form an antigen-antibody complex and thereby inactivate the potential pathogen, the antigen. Naive B lymphocytes are a type of professional antigen-presenting cell. And what will happen here is that we are constantly, throughout life, generating naive B lymphocytes. The naive B lymphocytes then go out into the world, and they leave the red bone marrow and go into any number of germinal regions. And in those germinal regions, they sit and wait for a couple of weeks or so. And if during that lifespan of a couple of weeks, their transmembrane protein now, which is an antibody, this is specific for these naive B lymphocytes, they have a transmembrane protein that's an antibody. If this binds to an antigen, that antigen is going to be endocytosed, much as we saw for the macrophages, and presented again on an MHC2 protein. Again, these naive B lymphocytes are professional antigen presenting cells. And they will present this antigen now on their surface on MHC2s. If they then run into a T helper cell that can recognize that antigen fragment on their surface bound to the MHC2, then the T helper cell will talk to the naive B lymphocyte and tell the naive B lymphocyte to make thousands and thousands of copies of itself. This is something called clonal expansion. And we call it clonal expansion because one of the interesting characteristics of the humoral branch of the immune system is that these naive B lymphocytes will randomly generate a protein that will become an antibody. It's not a transmembrane protein. And then that randomly generated protein will go out into the world and look for, sit and wait, to bind an antigen that's foreign. And if it binds that antigen that's foreign, then that single cell that has bound to the antigen will then proliferate. And it will proliferate from a single common cell, generating thousands and thousands, millions and millions of antibodies, which are all identical based on the structure of the antibody that initially was a transmembrane protein on the naive B lymphocyte. But none of that can happen without the naive B lymphocyte presenting to a T helper cell and the T helper cell releasing cytokines, basically local hormones that will tell that cell to divide. Then I'll tell you the story of the T killer cell, how in the T killer cells that are virus infected, those cells will be displaying foreign proteins on their surface. And those foreign proteins will be bound to MHC1, and the T killer cells will use their T cell receptors to recognize the foreign antigen, the antigen fragment on the MHC1, and that recognition then will tell the T killer cells to release perforins. It says the name perforins because it's going to perforate the membrane of the virus-infected cell and kill it. So we'll walk our way through that and see how that gets done. But let's start here with the idea of the macrophages as a professional antigen presenting cell. But we'll begin with the idea that that monocyte has arrived at the site of bacterial proliferation. It's matured in about 72 hours or so into a proper macrophage. And remember, the macrophage is an endocytic cell. And there will be proteins out here that can bind to either the bacteria themselves or a bacterial fragment. So I'm going to label this as a fragment of bacterial protein. Now recognize we could do the same story by using these as whole bacteria. But the idea is the same as we've seen so many times before. The binding of this protein to the transmembrane protein on the macrophage initiates endocytosis. The membrane begins to invaginate, a vesicle is formed, and these proteins, which can be quite large, inside the vesicle, are then targeted to the lysosome. Remember that the lysosome is a vat, an intracellular organelle, but it's really a kind of vat of proteolytic enzymes. So that when the vesicle carrying the fragments, or in fact the whole bacterium, arrives at the lysosome. In the lysosome, these bacteria or bacterial fragments are reduced, are digested in the lysosome into fragments known as antigen fragments that are about 16 to 20 amino acids long. So these are in fact just fragments of the proteins, because remember, most proteins are going to be several hundred amino acids long. And so these are just fragments of those proteins. And then the membrane will bud off from the lysosome. And now it's carrying these fragments, I'll continue to put them in red, these fragments of the original protein, bacterial protein. Then there's another membrane system in here called an endosome. It's kind of like a lysosome, but it doesn't have nearly as many of the degradative proteins. It's not nearly as low pH. So it's more of a kind of normal environment. This structure now I'm going to call the endosome. When the vesicle containing now the fragments that have been rendered into these tiny bits fuses with the endosome, this vesicle fusion delivers antigen fragments to the endosome. In the endosome, there's a particular kind of protein, and I'll show you this in black. There is this protein in here called MHC2. MHC2 stands for major histocompatibility class 2 protein. Now, that's a mouthful, so we abbreviate it MHC2. Again, MHC2 is confined to professional antigen presenting cells. If we blow this up a little bit, what we'll see is in the endosome, so let me do it over here. So in the endosome, there's this transmembrane protein called MHC2. And the antigen fragment then will bind to the MHC2 protein. And I'm going to show you this as a kind of triangle here. And this is meant to be the antigen fragment. So if we put it into this smaller image here, one thing I want you to recognize is that we can now form a vesicle. A vesicle with the MHC2 protein and its antigen fragment. When this vesicle fuses the plasma membrane, I'm running out of room here, so I'll put it down here. The vesicle fuses with the plasma membrane, I'll show it in green to represent the idea that this is the former endosomal membrane. When that happens, so we've seen so many times before, now this MHC2 protein is now exposed on the outside of the cell. And it's exposed on the outside of the cell where it has attached to it this fragment of the bacterial protein. So what's happened here is the macrophage has endocytosed the bacterium or a piece of it, rendered it into smaller pieces in the lysosome, attached those smaller pieces in the endosome, budded off a vesicle with the antigen fragment attached to the MHC2 protein, guide that vesicle to the membrane, and have that vesicle, through exocytosis now, have this transmembrane protein as the MHC2 protein display the antigen fragment on the surface of the macrophage. This macrophage now leaves the site of inflammation, goes into the blood vascular system, wanders around and finds its way to some germinal region where it can now present these antigen fragments to cells of the immune system, particularly T helper cells, and in some cases maybe T killer cells. So any questions on that? Okay, so let me go over to the right-hand panel and divide our world into that adaptive immunity into B lymphocytes and T lymphocytes. You'll recognize this as the cellular branch with the T lymphocytes, and with the B lymphocytes, we're talking about the humoral branch. Now these B lymphocytes, we're going to see where they come from and how they mature in a little bit, but right now I want to tell you that B lymphocytes mature into plasma B lymphocytes. And that these plasma B lymphocytes secrete antibodies into the extracellular environment. So this side of the story, this is B lymphocytes. They secrete antibodies. The antibodies then will bind to antigen, and antibodies bind to antigen to form antigen antibody complexes. This is really nothing more than a receptor-ligand interaction we've talked about so many times before, but in this particular case now the antibody is going to bind to that foreign protein, that pathogen, and hopefully inactivate it. So that's how this branch works. This branch, the cellular branch, works through T lymphocytes, which are further divided into T helper cells, also known as CD4 cells, and T killer cells, known as CD8 cells. What these two have in common is they have a transmembrane protein that's called a T cell receptor. Now, for both the T helper cells and the T killer cells, the T cell receptor recognizes antigen fragments. Presented on MHC1 or MHC2 proteins of other cells. The MHC2 proteins are recognized by CD4 cells. The MHC1 proteins are recognized by CD8 cells. And we'll walk our way through this and see, in fact, how these cell types function and what they do in the immune response. Before we get to that, though, what I want to do is I want to show you a slide. And this is going to be the story of the humoral branch of the immune system. And I want to show you this slide because this is a fundamental idea in immunology. And this is basically a slide which could have been drawn about 1910. What it shows on the ordinate is basically antibody concentration in the plasma. So these are antibodies that have been released by B lymphocytes. We didn't know that in 1910, but we know that now. Then this arrow here shows to the first exposure. This is the point at which the person or the animal has been first exposed to the foreign protein, the antigen. And then what we see here in this blue line represents the increase in the concentration of antibodies in the plasma. And you'll notice that it takes several days before we even begin to generate those antibodies and put them out in the plasma. So it takes several days after first exposure before we start secreting the antibodies from the B lymphocytes. Then as those B lymphocytes that are secreting the antibodies proliferate, then we increase the amount of antibody out in the extracellular environment. And it peaks, they show about 10 days here, sometimes as long as 14 days. That's why after you've been inoculated, say for flu, influenza vaccine, it takes about 10 to 14 days before you're effectively immunized. That's this lag in time, and we'll see why we're going to have to generate this response from a single cell. But you'll also know that one of the characteristics of the adaptive immune response is that it has memory, and that's also shown on this slide. So after some period of time and after the infection has been thrown off and we come back to normal health, we keep behind a very low level, it's called a titer, of antibodies in the plasma. And this represents a small a small subset of these B lymphocytes that were secreting antibody. We store them away. They're going to be called memory B lymphocytes. Those memory B lymphocytes are sitting in wait in all of your germinal regions. There may be a few tens of thousands of them. And as soon as they see that foreign antigen again, what they're going to do is they're going to very rapidly mount a secondary immune response and have a much more vigorous, a much more robust secretion of antibody. So this is the secondary antibody response. Two things to know about this. Here they show a delay of a few weeks. But in many cases, we can wait years, maybe most of a lifetime, before we are exposed to antigen again, and we then produce a secondary response very rapidly. The secondary response is more robust than the primary response. You see this, it's a higher level of antibodies in the humors. So these B lymphocytes have been ginned up. We have lots and lots of them, and they secrete the antibody out into the extracellular space for the secondary immune response. So that's the major idea of the generation of antibodies in the humoral branch and the secretion of antibodies into the extracellular environment. Well, with that, let's think a little bit about what we mean by antibodies. So over here, may as well go to the left-hand panel. On the left-hand panel, what I want to talk about is the structure of secreted antibodies. And I'll take this panel and I'll label it as the structure of secreted antibodies and their specificity. When we look at the structure of an antibody, we see that it has this common shape, and you'll see this commonly. It's this Y. This Y is composed of two identical heavy chains. So we've shown these two heavy chains in black. But in addition to the two heavy chains, out here in the short arm of the Y, there's another chain called the light chain. So the antibody has two identical light chains and two identical heavy chains. They're exactly the same, one to the other. Now, as a detail, these two branches of the Y will be held together by disulfide bonds. But in addition to those heavy chains and light chains, we can divide these chains into subregions. So out here in green, I'm going to call this HC. What HC stands for is the constant region of the heavy chain. If you take an immunology course, you'll see that this region, these constant regions, are divided into groups. There's IgG, IgM, IgE, and IgA. Well, we're only going to deal with something here called IgG, small g. This is immunoglobulin G. This is the type we see mostly released from B lymphocytes. This is one we're going to emphasize. And it will always have the same heavy chain. It will also have a region up here on a light chain called the HL region, which is the, I'm sorry. And this is the light constant region. So this light constant region will be the same from antibody to antibody. So these will always be the same. Where we begin to see a change, and I'll put this out here in orange, is out here at the short arm of the Y, out here. For the heavy chain, this is called the VH. This is the variable heavy region. And out here is the variable light region. Now, this two-dimensional image doesn't do justice to this protein. It's a proper three-dimensional shape. And it's out here at this short arm of the Y, out in this region. Remember, these variable heavy and variable light regions will be the same on both these short arms. This region out here is the site where antigen binds. The antigen is going to bind at this end of the short arm of the Y in the three-dimensional shape, which is provided by a combination of the three-dimensional shape of the variable heavy region and the variable light region. Let me show you that in a pretty picture. They show it differently here. So in lavender, that's the heavy chain, identical on both sides. In blue is the light chain. Again, these are held together by these disulfide bonds. Out here at the end of the short arm on the Y is the variable heavy and the variable light region, and this is the region that gives the three-dimensional shape that's specific for the antigen. And over here is variable heavy and variable light, giving the three-dimensional shape that's specific to the antigen. The light chains, again, are divided into the variable light and the constant light, and the heavy chains are divided into the constant heavy, which is this whole region, and the variable heavy, which is out here. And it's the blue shaded region where we have the three-dimensional shape where we can bind to antigen. Now, if we think about this for a little bit, what I want to show you here in this slide is briefly describe how the formation of the antigen-antibody complex can now inactivate these foreign or potentially pathogenic proteins. Most commonly what we see is there's a kind of masking or neutralizing, and we'll now cover over the potentially pathogenic sites on the foreign protein. Now, for example, this could be a virus. So this could be the COVID virus. And we can have antibodies now that will bind specifically to the spike protein on the COVID virus. And by binding to that spike protein, which remember has to bind to the ACE2 receptor, by covering over that transmembrane protein, the extracellular region of the transmembrane protein, with the antibody, we can prevent the virus from binding to the ACE2 and prevent this virus from entering the cell. That's how we think these antibodies from the humoral branch of the immune system are going to function. In other cases, because these antibodies are Y-shaped, they can cross-link proteins. They can even cross-link viruses. And in so doing, by cross-linking them, they then lump them together so they're more easily gobbled up by macrophages. And the macrophages then can destroy them. And in some cases, if we have free proteins out and about, we can cross-link the proteins and also precipitate them, allowing them to be gobbled up by macrophages. Over here is complement fixation. You've taken immunology course, you'll spend a lot of time on complement fixation. We're not going to deal with it here. Just recognize that what we have are these antibodies that are secreted into the extracellular environment and that they are then priming the system to destroy those foreign proteins. This next slide is a cartoon, but I like this cartoon because it gives a lot of information in a small number of points. This red structure is meant to be the antigen. This could be a protein, a large protein. And then we can see that this large protein can have a number of sites on it to which it will bind antibody. The antibodies are recognizing short segments of protein, no more than 16 or 20 amino acids long. And that specific region of 16 or 20 amino acids in an individual protein, this particular region of 16 or 20, can be recognized by this antibody shown with having a particular shape out here. Another region having a slightly different three-dimensional shape can be bound by this antibody, but this is a different region of the protein. And another one up here with a slightly different shape bound to another specific antibody. So a given antigen can have a number of antigenic sites to which we can make antibodies. And if you look at the literature on COVID, you'll find that the spike protein is the one most commonly where we make antibodies to it, but we make antibodies to a whole host of different places on that spike protein. And what the pharmaceutical companies are trying to figure out is what's the most attractive site to generate a vaccine with that particular amino acid sequence that will be most effective in generating antibodies that can protect us against the virus. So any questions up to there? Okay, so that's the story of secreted antibodies, and we're running out of time. Let me come back here and tell you the story here of the selection of B lymphocytes. So we'll take this panel and we're going to label it as clonal selection of B lymphocytes. I want to tell you where these B lymphocytes come from and why there's this delay of some time between initial exposure to the antigen and the generation of antibodies. Remember, that delay is the period of time when the innate immune system is mounting a holding action to hold those pathogens at bay until we can generate a proper antibody response. And that proper antibody response now is going to be initiated by a cell type called a naive B lymphocyte. Naive B lymphocytes and clonal selection. So I'm going to start here with a naive B lymphocyte. This is a naive B lymphocyte. You'd find this in the red bone marrow. And we're generating these all the time. And I don't have time to tell you the exquisite story of how we use the genome to make one particular protein and how we can make thousands and thousands of different proteins and potentially protect ourselves from an almost infinite variety of potential antigens. But what I will tell you is we start out with a transmembrane protein out here that looks like an antibody. This is a transmembrane protein of an antibody that's on a naive B lymphocyte, and the naive B lymphocyte now goes out to a germinal region. And in the germinal region, it sits and waits with its antibody out here. Now, if that antibody binds to a protein, it binds to a protein here, which is in fact going to be an antigen. Just as we saw on the macrophage, this initiates the process of endocytosis. We package this protein into a vesicle. With the antigen attached to it, just as we saw for the macrophages. We send it off to a site here. I'm going to change colors here because my plasma membrane's blue. We send it off to a lysosome. And in the lysosome, this antigen, this foreign protein, is now digested. The antigen is digested into an antigen fragment. It's then passed to an endosome. And just as before, this endosome contains MHC2 proteins. So this is MHC2. Remember, we said naive B lymphocytes are one of the classic professional antigen-presenting cells, so therefore they have MHC2. In the endosome, we attach a piece of this protein that was originally bound to the outside. It's now then in the endosome trafficked to the extracellular site, fuses. So now again, just as we saw in the macrophage, it's the same story. We have this transmembrane protein, and attached to it is the antigen fragment. So this is the antigen. Now on the right-hand panel, I'm going to now describe the interaction between that naive B lymphocyte with its MHC2 protein out on its surface and its antigen fragment interacting with a cell called a T helper cell. So we're going to take this cell, and I'm going to blow it up and simplify it to the point where we left off. So this is the naive B cell. It has out on its surface the transmembrane protein called MHC2. The MHC2 protein in this case is displaying this fragment of the foreign protein it bound. So this is the antigen fragment. Now, in black, there's another cell. This is the T helper cell. It's also known as a CD4 cell. And one of its characteristic transmembrane proteins out here is this transmembrane protein, I'll put a little dot on the end of it, called CD4. And the CD4 binds to the CD4 binding site on the naive B lymphocyte. So it binds to this CD4 receptor, but also on this T helper cell is a protein. This protein comes as an alpha and a beta subunit, which now make up the T cell receptor. If the T cell receptor now recognizes this antigen fragment, that mobilizes vesicles here. The vesicles fuse to the plasma membrane, and they now release molecules called cytokines. On the naive B lymphocyte, there are receptors here. To which we can bind the cytokine. This is usually something called interleukin 4, sometimes 8. There are like 64 different interleukins. There are a whole flock of them. They do a whole bunch of different things. If you take your immunology course, you'll drill down into these. I just want to tell you that these interleukins are a type of cytokine. Cytokine binds to the receptor, and that binding then causes cell proliferation. So these B lymphocytes, these naive B lymphocytes, start to divide and make thousands and thousands and thousands of copies of themselves. And all of the daughter cells of this naive B lymphocyte will contain the same transmembrane protein that we started out with. It's a transmembrane protein. And after a few cell divisions, those transmembrane proteins, which are the antibodies, are packaged into vesicles. And when the vesicles fuse to the plasma membrane, those antibodies are now released. And that's the way we go from having a single cell with a transmembrane protein as an AB lymphocyte, through its interaction as a professional antigen-presenting cell bound to the T cell receptor that now initiates cell division in these cells to make copies of it, to eventually convert this cell into a plasma B cell. Okay, with that, what I'm going to do now is I'm going to hold you to anything I said in the introduction. And so you don't have to listen to any more of this. So I won't ask any questions on the exam beyond what I said in the introduction. But I want to tell you the story. I want to tell you the story about the T killer cells, because I just think it's a fascinating story, and I can spend a moment here so that if you want, you can spend some time with the details of this. And you can see how, in fact, the T killer cells are monitoring for virus-infected cells. So we could use the story of COVID, and I'm sure we're going to find that we're going to generate T killer cells that will seek out and destroy virus-infected cells. So I want to spend a moment here telling you how this happens, even though we're beyond time. And again, if I haven't said it in the introduction, I won't hold you to it. But let's say out here, we have a cell. I'm going to label this as a somatic cell. Somatic cell is any nucleated cell. So this is any cell in your body. This is any cell other than an erythrocyte. And what happens is that throughout life, within the endoplasmic reticulum, so this is meant to be the ER, there are proteins that are taking fragments. They're taking usually parts of the cytoskeleton. And they're chopping them into 16 to 20 amino acid segments. They move these proteins, I'm going to show it as a dot, across the membrane. And in the ER, they bind these protein fragments to a protein called MHC1. Again, this is major histocompatibility class 1 protein, and it's on all cells that have a nucleus. So what's happening is we're moving this protein into the lumen of the ER, and when that happens, we're now going to attach that protein to the MHC1 protein. I won't go through the rigmarole because we've been through this so many times. We'll then bud this off as a vesicle. This particular vesicle is now going to contain the MHC1 protein. And it's going to process it through the Golgi apparatus. And finally, a vesicle will bud off, and it's going to contain this MHC1 protein. And attached to the MHC1 protein is going to be this fragment of one of your very own proteins. When this vesicle fuses with the plasma membrane, we're now going to display the MHC1 protein on the cell surface with this bit of self-protein. So I'll redraw that over here so I can have access to it. So here's our MHC1 protein. And it has out on its surface this antigen fragment. Now, in green, there's going to be another cell. And this is going to be a CD8 cell, also known as a cytotoxic. T killer cell. These T killer cells are always on the prowl. They're out on maneuvers, and they're always out on patrol. And what they're looking for are MHC1 proteins. And again, just as we saw on the T helper cell, we have this alpha and beta subunit of the T cell receptor. Almost always, when this alpha and beta subunit of the T cell receptor check out the antigen fragment, nothing happens. Oh, one more point I have to make here. There's a transmembrane protein here, put it in red. This transmembrane protein is called CD8. Just as we saw for CD4, which bound to the MHC2 protein and held the cells close enough so that the antigen fragment could be explored by the T cell receptor, the same thing happens here. We moor the cells together, we bring them close enough so that the alpha and beta subunits can see if there's a three-dimensional fit between the antigen fragment and the T cell receptor. Almost always, 99.999% of the time, there's no fit. Because there's no fit, the T killer cell lets go and wanders off and goes to another cell. But now and again, we have a virus-infected cell. And remember when we talked about viruses, when we talked about the CoV-2 virus will generate proteins. It's basically taking over the protein synthesis machinery of the cell. So it's going to make, a virus is going to make viral protein. That viral protein then will be brought into the endosome just like the ordinary cell protein was. But now we're going to chop it into an amino acid sequence of about 16 to 20 amino acids, take it through the same circuit, and express it on the outside. Now I'm going to change the antigen fragment here from a circle to a triangle and say that this now is the antigen fragment of the viral protein, and now we're going to have a fit. We're going to have a good three-dimensional fit between the alpha-beta subunit of the T cell receptor and the antigen fragment. If there's a good fit, as before, we're going to now mobilize vesicles in this T killer cell. They're going to come to the surface, they're going to fuse the plasma membrane, and now these vesicles are going to be full of a protein called perforin. The perforin doesn't seem to intercalate itself into the T killer cell, but what it does is intercalates itself across the membrane of this virus-infected cell and generates calcium channels. The influx of calcium in massive amounts induces the apoptotic machinery of the virus-infected cell and kills the virus-infected cell. So that's how, when you have virus-infected cells, so imagine you or one of your friends has COVID. What they're doing is they're generating lots and lots of CoV-2 proteins, and the CoV-2 proteins in their cells are generating CoV-2 proteins that are, some of which are taken into the endosome, passed through the system, placed on MHC1, and inspected by the T killer cells. If you've ginned up a series of T killer cells that can recognize that fragment, then you'll kill that virus-infected cell. And again, what my suspicion is that because of our thymuses, as we age, they're not nearly as effective. Because as we get older, this response is less effective because we simply don't have as many T cells to choose from that will recognize the antigen fragment and thereby kill the virus-infected cell. So I'll leave it at that. And again, I won't hold you to anything on the T killer cell response except what I said in the introduction.